So I got back to my office after lecture today and realized that for some reason my Zoom recording was only 5 minutes long. So I'm going to re-talk through the lecture. I have my slides here and what I wrote in on them during lecture, so I'll try to simulate exactly what I said in lecture. And because there's about 10% of you that are either testing positive or in quarantine. So I know that there's quite a few people that couldn't make it to lecture today. So again, I'm trying to figure out what I did wrong, but I'm going to record it again this way, and hopefully it's similar to pretty much the same. Let's see, I did post an announcement in the announcements on Blackboard, a link to the office hours that I'll be having by Zoom every Friday from 9 to 11. So stop on in if you want to see me without a mask on, or you just want to say hello, or if you have any questions. Let's see, what else? You can be working on Chapter 1 homework. We finished that chapter, so if you're not already got your code and into mastering, make that a priority to get that done. Hopefully, remember your first introductory homework assignment is just 2 questions. Hey, that basically gets you to get signed up into mastering. That's supposed to be due on Tuesday. If you're having trouble, any technical problems, or you haven't been able to purchase that yet, just send me an email and I can reset that. Again, little 2-question assignment. Otherwise, all your homework assignments are due the day of our first exam. So in general, you have a little time, but hey, you've got a long weekend, get started, get that first homework chapter 1 out of the way. After you listen to this lecture, you can also do chapter 4, and part of that first part of chapter 6. Okay, we didn't get to chapter 11 yet. All right, so last time I talked about where we finished. I was talking about a pure culture. Remember, that means when you only get one kind of organism within the petri dish, within the culture, or within the test tube. Okay, and we keep those cultures pure by aseptic technique. And typically, if you're in lab, you would have been doing the aseptic technique lab either today or yesterday. We just haven't got that lined up as good this semester. Okay, so just finishing up culture techniques. If you don't have a pure culture, then you've got more than one species in one, say, petri dish or test tube, and we call that a mixed culture. All right, so here's a great picture of a mixed culture. If you were to take any sample from the environment, say scrape the bottom of your shoe and streak it out on a plate, you're going to get lots and lots of different organisms. And when you streak on solid media, that's pretty easy to see because we've got a variety of different colors and shapes and textures that bacterial colonies tend to grow. So here we've got at least 12 different things growing on this plate. We like to keep our cultures in lab pure, usually. We like to work with one organism at a time, especially if we're trying to identify it. We want to make sure it's all the same thing, or else we're going to get conflicting results. Okay. I want to talk about some kind of categories of different culture media to kind of understand. I'm giving some specific examples of different kinds of media, but we have some broad categories that's helpful to understand. All of our microbial media that we use to grow colonies up, grow cells up, is either going to be considered defined or complex. Okay, with a defined media, that means that the exact chemical composition is known. So here's an example of a defined medium. Don't memorize this recipe. This is actually a recipe for minimal media, and we would streak organisms out on this kind of media if we were trying to grow up an organism that was able to synthesize most everything that it needed to grow. Organisms that fix their own nitrogen, they grow fine on minimal media. But lots of times, most other different kinds of organisms wouldn't grow very well. But if you look here, we can see usually you've got, the formulas for what's in there. We know exactly how much of each of these ingredients that's in that media. A complex media, basically, we don't know the chemical composition exactly because the nutrients are going to come from some partial digestion of beef, soy, or yeast. Okay, so a complex media is always going to have beef extract, soy extract, or yeast extract in it. Right, so here's an example of a complex media, and it's one that we use quite a bit in lab. It's a general all-purpose media called nutrient agar. So I wanted to look a little bit more carefully what's in the nutrient agar. Peptone, that's just a source of amino acids. And of course, organisms use amino acids as building blocks to make their own proteins, or in some cases, they can use that as an energy source. Here's what makes this a complex medium, that either beef extract or yeast extract. That's just a slew of nutrients. It's been digested by some enzymes. So it's going to release carbohydrates and vitamins and minerals, lots of things that cells need for energy and growth. If you see agar in a medium, that just means it's a solid medium. So we put agar in there as a solidifying agent. Remember, agar is not a nutrition source. In the agar medium, there are other things that, like we see here, that provide nutrients for the organism. And a little pinch of salt. Again, don't memorize amounts here, but this is just a little bit of salt. It's not to make it taste good, but in order so that we have osmotic balance. And we're going to do a little review of osmosis next week, but this makes the medium pretty close to isotonic, so that we don't want our cells, losing too much water or gaining too much water. So we want to keep them happy. And that's, pretty close to physiological salt concentration for the organisms. Okay, we add a certain amount of water, and then typically for this medium, we pH it to about neutral. Okay, so all medium is either considered defined or complex, and again, it's this presence or absence of these extracts really that make the difference there. Media can also be either selective or differential. It can be neither one, or it could be both. Okay, selective media is media that has something in it that's going to inhibit the growth of some organisms. So oftentimes that's an antibiotic that will keep certain organisms from growing, but it can be other things like a high concentration of salt that would select against a lot of organisms, or maybe something else like pH. So here's an example of a selective medium. This is from your textbook. It looks like they probably took a sample of soil. I've taken soil samples before, and it looks a lot like this plate here on the left. So they probably took a soil sample and put a soil suspension maybe on top of some. This is nutrient agar over here, and you can see you've got a whole slew of different colonies growing here of bacteria. Sabarodes agar is a media that's almost exactly the same as nutrient agar, except Sabarodes agar is pH'd to be a little bit lower. And that lower pH actually selects against bacteria. So it favors the growth of fungi. Okay, fungi are more likely to grow at lower pH. So you can see these fuzzy colonies here, and that's indicative of a fungal colony. All right, so just changing the pH there will select against bacteria. Sometimes we get some fungal colonies here on nutrient agar, but they often kind of get outcompeted by the bacteria. Okay, you're going to work with some selective medias in lab. Again, the selective agent is an antibiotic that keeps certain kinds of growth from growing. Differential media, everything grows on differential media, but certain kinds of growth will take on a different appearance. Okay, and that helps us, it tells us something about the organism and helps us put it in a group. So, for example, sometimes that differential agent will be a pH indicator and maybe a sugar. So if you add a sugar into some medium, organisms that can utilize that sugar will always give acidic end products. So you have a pH indicator, and then you're going to have a color change, and that color change will tell you, oh yeah, that organism can use lactose. Okay, it tells us, gives us some important information about the organism. So here's an example of some media that's considered differential. You're going to be, if you're in lab, you're going to be inoculating blood agar later on in the semester. And here's 3 different organisms streaked out on blood agar, and you can see their growth has a different appearance. Some organisms totally lyse red blood cells. Okay, you see like a yellow clearing here. Some organisms only partially lyse red blood cells. What that means is they don't break down the hemoglobin. So the hemoglobin stays intact, and that's why you get a little bit of a greenish color here for that partial digestion. And some organisms aren't able to lyse red blood cells at all. All right, so we can tell something physiologically about the organism based on how their different appearance. So that's really helpful when it comes time to try to identify organisms. Okay, if you were to take blood agar and add a certain antibiotic, okay, now this media is selective and differential. Okay, in lab you'll be adding an antibiotic that keeps certain organisms from growing. So a media can be one or the other, both. Some medias like nutrient agar, it's not selective or differential. It's true that you might see colonies that look a little bit different here on the nutrient agar, but it doesn't tell us anything like nutritionally or physiologically about the organism. So that's neither, nutrient agar is neither selective nor differential. And I just added on the end here, some medias are anaerobic. So if we're trying to grow an organism that can't live in the presence of oxygen, then we need to keep that away. Sometimes we can do that by the use of certain medias. Oftentimes we have some kind of physical thing like a glove box or something that would keep the oxygen out. But you are in lab in a few weeks going to be inoculating thioglycolate broth, which is the whole point of that broth is to keep oxygen out. So I thought I would mention that here. Thioglycolate is actually a reducing agent. So essentially what it does is reduces any free oxygen in that media into water. So we've got an indicator in here that tells us if there is some oxygen. Of course, some diffuses in under the cap. But essentially below that, because of that thioglycolate throughout the medium, there's very little, if no oxygen. So if we're trying to understand the oxygen requirement of an organism, we would inoculate that in there and look to see where it grows. Okay, if it only grows at the top, then it must be an aerobe. If it only grows at the bottom, then it must be an organism that's poisoned by the presence of oxygen. And lastly, as we think about culturing techniques, just remember there's lots of organisms that we have no idea how to culture in the lab. And of course, there's lots of organisms we don't even know about yet because we haven't been able to culture them in the lab. For instance, Mycobacterium leprae, that's the causative agent of leprosy. The only way we have to grow that up is in armadillos. I imagine that took a lot of trial and error to figure that out. But we don't have any medium that we can get that organism to grow up on in the lab. Likewise, the organism that causes syphilis only grows in rabbits. Okay, we haven't been successful in growing that up in the lab. And of course, viruses take special culturing techniques that we refer to as cell culture. So basically, there's a layer of some kind of cells, usually some kind of eukaryotic cells, usually that we have to grow a layer of in order to get the viruses to grow up inside those cells. So you see some little different kind of, weld containers when you're trying to grow up viruses. All right, moving on to chapter 4. Again, I know we're skipping around here a little bit, but typically we would be starting microscopy pretty soon in lab, so it does match up a little bit better. And we've got some overlap between lab and lecture here. Two fairly different topics we're going to be covering here, microscopy and taxonomy. The taxonomy slides are a little bit dry, a little boring, but important information nonetheless. So we're going to start talking about some general principles of microscopy. We're going to think about, well, what do we need to do in order to see our cells the best that we can see them. You'll get that in lab as well. Good to repeat it here a couple different ways. And then we're going to talk about some different kinds of microscopy. So in lab, you're only going to do one kind of microscopy. There's a lot of other interesting things out there. I'm not really going to address staining. There's some staining. They talk about it in your textbook, and I've left some homework questions about staining, and I've marked them just as practice, just because that maybe will help you with that lab a little bit. But you can just skip those if you want. The staining procedures that we are skipping in lab this semester, I am going to talk about a little bit more thoroughly than I normally would in lecture. So hopefully you'll still get exposed to those, and we'll get to those next week, most likely. And then a little discussion of taxonomy, some terms, and the contributions that Carl Linnaeus, who we mentioned last time, and Carl Woese have made in terms of our classifications there. All right, so first a word about units. So of course when we measure things, we're going to be using metric, which is a little bit unfamiliar to most people, and we're going to focus on, of course, the tinier units of the metric system, which most of you probably haven't used very much. Okay, so of course the meter is sort of like the basic unit of length in the metric system. The next, if we move the decimal place three places, here we go, 0.001 meter, that is equal to a millimeter. And of course you can see a millimeter with the naked eye. A really big protozoa, perhaps you could see with your naked eye almost. Okay, so unaided human eye. Okay, but we get any smaller than that, and we're going to start to need probably a light microscope. We move 3 more decimal places to the left, 1 times 10 to the negative 6 meter. That unit is equal to a micrometer, or sometimes called a micron. And that's a really important unit in terms of measuring bacterial cells, because the typical bacterial or archaeal cell is about 1 micron in length. There's lots of variation, but that's kind of a good standard to go by. And organelles in eukaryotic cells are probably going to be a little bit larger than that. And here's a eukaryotic cell, maybe on the order of about 10 times larger than a bacterial cell. Okay, so we can see those with a light microscope, but basically if we get any smaller than a cell, like if we're trying to look at structures of cells here, we're going to need an even smaller unit, 3 more places to the left, 1 times 10 to the negative 9th meters, that's a nanometer. Okay, so structures like ribosomes in cells, pretty much the only way we can see those is by some kind of electron microscopy, and we'll take a look at those in a minute. Okay, so there's a couple homework questions in your book that ask you to kind of convert between nanometers and micrometers and millimeters. It's just a matter of thinking through which way to move the decimal place. And if you know that the nanometers is the smallest, and then the micrometer, and then the millimeter is the biggest, then hopefully that will make a little bit more sense. All right, so if you have me for lab, this is going to sound very similar, again, when you hear this in lab in a few weeks. But it's important to understand, again, these kind of big overall themes and how we're going to see our cells very, very clearly. Okay, we have to think, first of all, about magnification. So obviously you need lenses that are going to magnify what you see. They're going to increase the size of what they really are. And typically with a microscope like what we use in lab here, these are bright field microscopes, typically we can increase magnification up to about 1,000 times. We could actually make lenses to increase the magnification even more than that, but it really wouldn't do us any good, because once we get larger than about 1,000x, the properties of wavelengths of light start to limit how clearly we can see something. So then we have to understand a little bit about resolution. Okay, resolution can be thought of as a measure of clarity, but it's really more that it's not just that your cells are in focus. It's a measure of whether we can tell whether objects are separate from each other. All right, so the ability to distinguish two objects as separate under the microscope. So if we look at a bacterial cell under a light microscope, the cell wall and the cell membrane, basically it looks like one structure. We know that they're separate, but we don't have good enough resolution to see the cell wall as being separate than the cell membrane. You could think about it in terms of a telescope as well. If you use a telescope to look at a couple of stars that actually rotate around each other, you might not have good resolution to tell that they're actually two separate stars. It might look like just one star from your telescope. All right, so we've got a formula here that kind of helps us understand how we can improve resolution. All right, so I think this is I know it's in your lab book, and I'm pretty sure it's in your textbook too. So resolving power is equal to the wavelength of light, so the length of the wavelength of light divided by the numerical aperture. And what you need to understand first is that good resolving power is going to be a low number, because this is actually the distance that two objects need to be separated from each other until still be able to tell that they're separate. So if they're really, really close together and you can still see that they're separate, that's good resolution. If they have to be far apart, then your resolution is not so good. All right, so two things that we can monkey with a little bit, wavelength of light and numerical aperture. Let's start first with the wavelength of light. If you're going to improve your resolution, one thing that you can do is shorten that wavelength of light. Let's see. So visible light has a wavelength of between like 400 and 700 nanometers. If you were to use only blue light, that's the shorter wavelength of visible light. In theory, say if you had a blue light down here, in theory that would give you a little bit better resolution, because the smaller you can make the numerator, the smaller this whole fraction would be. So, right, 1/3, for example, to throw some numbers in here, is a smaller number than 2/3, right? So if you can get the wavelength shorter, you're going to get better resolution. And so if you use electrons, which have a wavelength very much shorter than visible light, that kind of demonstrate why electron microscopes have such amazing resolution. Okay, the other thing that we can look at here is the numerical aperture. And what the numerical aperture is, it gets complicated in terms of the physics of light, but we can define it as the range of angles of light that are accepted into that microscope lens. Okay, and if we use a special kind of lens, an oil immersion lens and oil, then that's going to increase the numerical aperture. All right, and if we increase the denominator, so 1/4, right? This is a smaller number than 1/3, so that's going to give us better resolution. All right, so this figure demonstrates that the whole angle of light thing. Okay, so here's, if you're using low, well, what do they call it, scanning, medium, or high power on the microscope in labs, those objectives are designed to not actually be touching your cells. We don't usually use a cover slip, so I don't know why that's thrown in there, but it doesn't hurt anything. Typically, the light comes up from the bottom. When it passes through the glass and then hits the air, it typically bends off into lots of different directions, and some of those go up into the light source. But if you use a lens that's designed to be used with immersion oil, that's going to be the 100x lens on the scopes in lab, that will not focus on your cells unless it's touching the immersion oil. So you typically in lab, you're going to find your cells on low power, and then you're going to add a drop of immersion oil. Again, we don't usually use a cover slip. And then the 100x lens, the oil immersion lens, you'll lower it right down into that oil. And because immersion oil has the same refractive index as the glass, the light doesn't bend as much when it changes medium. So we get more angles of light actually ending up into that objective lens. So essentially that increases the numerical aperture. Okay, one other thing we need to think about when we're trying to see our cells clearly, and that's, do we have good contrast? Because you might have great magnification and great resolution, but if your cells look the same as the background, you're not going to be able to see them. Okay, so contrast, the ability of cells to stand out from their background. Here's some cheek cells that probably have just been thrown in a wet mount and looked under bright field microscopy. If you were really skilled, you could find those, but it would be probably very difficult. So typically what we do to increase contrast, number one, we stain the cells. So here's some cells that have been stained with something like methylene blue probably. You can see the nuclei of those cheek cells. All these little dots that you see out here, those are bacteria. So that gives you an idea how much smaller bacteria are than a typical bacteria to a typical eukaryotic cell. Okay, one other thing we can do to improve the contrast here, if you aren't using stain, then if you dim the light a little bit on a bright field microscope, sometimes that will enable you to see the edge of the cell a little bit better. And of course, we have some kinds of microscopy that I'm going to talk about in a minute that you don't have to stain the cells in order to see them. But with a bright field scope, like what's the most common microscope to use, dimming the light will help a little bit. If you turn the light up, that just bleaches everything out. So dimming it will help a little bit and often enable you to see the cells if you're good at finding them. Okay, so these are the kinds of microscopy that we are going to go through here in class. They're kind of the most famous kinds. There are a few different kinds in your book that we're not going to get to, like atomic force. So we're going to go through these one by one, and I'll show you an example of what your cells should look like, or an example of what some cells have looked like when we've used this kind of microscopy. All right, so bright field microscopy, and it usually has a hyphen there that I forgot to put in there. Again, this is the most common kinds of microscopy. This is what we use in lab. They're usually compound microscopes, meaning there's a series of lenses in them that enable us to get better magnification. I'm not so worried about the parts of a microscope. I'll let your lab professor, quiz you on that if they want to. What do our cells look like with bright field microscopy? Typically, there's a light background, like this example here. Sometimes we might stain the background, so that's not always true. It could be red or blue or whatever. But the light background, and then most often we stain the cells that we're trying to see. Like I said, we can get up to about 1,000 magnification with bright field scopes, and our resolution is going to be on the order of about 0.2 micrometers. If you converted that to nanometers, okay, it's going to be about 200 nanometers, okay? And mostly I bring that up because I want to compare that later on to the kind of resolution that we see with electron scopes. So if you're in lab, that's the only microscopy that we're going to use. Still very common are dark field microscopes, and this diagram down here is a little bit confusing. Dark field scopes employ what's called a dark field stop. So this right here is actually turned on its side, and it's probably going to be like right here, probably on top of the condenser in that microscope. Essentially, that stop keeps light coming in. It can't come in a straight pathway, so it can only come in at kind of an angle to see your cells. Apparently, you can make a bright field microscope kind of act like a dark field microscope by putting a quarter on the light source. Okay, so again, light can't come straight up through there. It has to come in at an angle. And because it comes in at an angle, basically it's only the edge of the cell that is illuminated. So the cells appear light and the background is dark. The advantage here is that we don't have to stain cells in order to get good contrast. So we can look at cells when they're alive, and so then they're moving around. Staining always kills our cells. Okay, phase contrast microscopes. There's two different kinds of phase scopes, and I did include this diagram here. Your new edition of the book doesn't keep it. It just kind of skips over this. It gets a little bit, again, into the physical properties of light, which I don't totally understand, so I don't really want to go there too much. But the basic idea is that if light rays are in phase, you get a little brighter image. And if light rays are out of phase, meaning your troughs match up with your peaks here, you get a little bit of a darker image. So when you have light rays passing through, say, your bacteria here, the light rays that pass through there are going to get knocked out of phase by just a little bit. And then phase microscopes include a phase plate that is going to knock it out even a little bit more, so that then we can see kind of the edges of this bacterium a little bit easier, again, without having to use stain. Okay, so some contrast is created because those light waves are out of phase. So because we don't have to stain them, we can view our cells when they're still alive. So that's a big advantage. And I'll show you an example of what this looks like in a minute. There is a second kind of phase microscope, and it's called differential interference contrast, or DIC for short. That's also known as Normarski microscopy. And again, this is a type of phase contrast microscopy. The light that's used is somehow the optical path is a gradient of the optical path, so I don't totally understand that, but we get a little bit more of a 3D view with this differential interference contrast. So if your book has a nice comparison of those 4 kinds of light microscopy. If you're not staining cells, this is what looks like a cheek cell would look like. All right, so bright field here, very kind of difficult to get good contrast if you don't stain them, but again, light background. With the dark field, you get the really dark background, and then kind of the edges of the cell or whatever are going to look light. The phase contrast, gradients of gray here, we often see a little bit of a halo effect as kind of an artifact when we're doing phase contrast microscopy, but again, it's a real advantage sometimes to be able to see your cells when they're alive. And then Nowarski looks a little bit more 3D, so a little bit less of any kind of artifact around there, a nice, crisp image there. Okay, so LM, that stands for light microscopy. Always pay attention to your units here, and that sometimes can help you understand what you're looking at. Right. Also considered light microscopy is fluorescence microscopy. It uses light in the ultraviolet range. So I mentioned this before, between 400 and 700 nanometers, that's visible light. A little bit shorter than that is the ultraviolet wavelength. Okay, because that's a shorter wavelength, remember that's going to give us the advantage of a little bit better resolution. Okay, and we also use some special dyes that fluoresce when the UV light hits them. Now these scopes look very similar to the bright field scopes that we use in lab, but they do have some kind of plate when you look through the optical lenses. So obviously the UV light, you're not looking into UV light because obviously that's pretty bad for our eyes. So just a little bit more complex than kind of our regular scopes. Fluorescence microscopy is often used as a really important diagnostic tool. So especially in terms of diagnosing somebody that's ill with tuberculosis. It's a complicated disease, and it's often difficult to understand sort of at what point a patient is, whether they have an active case or a case that's not actively spreading. But often you'll take a sputum sample, and that's going to be kind of what a patient like coughs up from their lungs. And if you were to just look at that under the light microscope, it doesn't really look very exciting. You can't even really see any cells there. But if you add a dye that, we have a special dye that actually sticks only to that bacterium, a protein, actually mycolic acid in the cell walls of these organisms that cause tuberculosis. It's called auramine rhodamine. But that only sticks to the tuberculosis bacterium, when you look at it under the fluorescent scope, if that organism is there, then it shows up and fluoresces. So it's a really nice tool, again, in terms of diagnosis. On those same lines, if you use a technique called immunofluorescence, this is another way, again, a good diagnostic tool where we actually can tag antibodies specific for a particular protein, for example, tag a fluorescent dye with those antibodies. So we're going to talk lots more about antibodies and antigens later on in the course. But in this example, you've got an antibody that's selective, that only sticks to a particular protein in the cell wall of the bacterium that causes bubonic plague. And it's called Yersinia pestis. So if Yersinia pestis is in that sample, the dye with the antibody is going to stick to it, and it's going to fluoresce bright green. All right, getting a little bit more complicated now. Confocal microscopes are, again, a level up of complexity from fluorescent microscopes. They also use ultraviolet light, but they use ultraviolet lasers. And this is supposed to be, manufacturers of confocal microscopes advertise them as being sort of a bridge between light microscopy and electron microscopy. Okay, you get better resolution because apparently you have these lasers that are shining the light through a very small aperture opening, and then you have a computer that actually takes very thin sections and puts the image back together again. So you're viewing the image on a computer screen, and it's an image that's 3D, so you could move this around and look at it from all sides. This is a picture of some cow brain, I believe. Okay, and we have one of these scopes in Kinchi. I haven't looked at it, but it looks something like this here. We looked into getting an electron scope at Grand Valley several years ago, and it just made more sense to get a confocal one that was a little bit cheaper, that didn't require quite so much upkeep as what an electron scope would. So here's some images. I didn't post these, but here's comparison on the top here is what these images look like under a fluorescence scope. And here's comparing how they look under a confocal scope. So we get much better clarity here with the confocal. This first image is, let's see, I think it is human medulla cells. The second one is rabbit muscle fibers, and the third is a grain of pollen. So a little nicer resolution with the confocal scope. All right, and lastly, of course, we have to talk about electron microscopy. Okay, the big advantage here is in using electrons that, of course, the wavelength is much shorter than light, so we get much improved resolution over using any kind of light microscopy. Now, there definitely are drawbacks to this. The specimen preparation, whether you're using a transmission electron microscope or a scanning electron microscope, the preparation of the cells, I've heard researchers say, oh, it took me a month just to prepare the cells and to get it right. You usually have to go through a special method to fix the cells, and with any kind of electron microscopy, you have to view them in a vacuum because air is going to scatter those electrons. So you can't have them in air, apparently. So some special considerations there. Magnification, though, pretty amazing, 10,000 up to 100,000 times magnification. So good resolution, good magnification. What's the difference between a transmission electron microscope and a scanning electron microscope? The transmission gives us the best resolution, and that's about 3 nanometers. Remember, our bright field light microscopes, the resolution was about 200 nanometers. So, pretty, about 3 orders of magnitude different there in the resolution. With the transmission, the specimen prep is especially long and involved. You have to coat the specimen with some sort of plastic, and then you have to get it in thin sections. So you use something called an ultra microtome. If you picture, like if you've ever watched them in the deli using their blade to cut off slabs of meat, okay, think that only on a much different scale, like extremely thin sections. Okay, and then the electrons are going to pass right through those thin sections. So what we see is a cross section. I'll show you an example of that in just a minute. Compare that to a scanning electron microscope. The slices are not thin. Basically, we coat the cells in typically a heavy metal, such as gold. So it's still kind of difficult to prepare the sections, but You don't have to slice it as thin, so typically what we see is a 3D image and not a cross-section. And so the resolution is not quite as good as the transmission electron micrograph. Okay, so here's some examples. Again, the transmission electron micrograph, and if you're never, these images always should be labeled as to what kind of microscopy here. So TEM, that stands for transmission electron micrograph. And notice the units here. This is going to be much smaller than what the units were for our other kinds of microscopy. Oftentimes the pictures are, or the micrographs are artificially colored. All right, so they might just go in and color that just to help people to kind of maybe understand when one structure is separate from another structure, because it's really easy when you look at cross sections like this to like have no idea what you're looking at. It takes some training, and there's always some artifacts involved, so it's kind of hard to know, well, what's a real structure and what's just some kind of artifact of electron microscopy. The scanning electron micrograph pictures are the ones that are often much more interesting in my mind because, again, they're 3D. This is a marine algae, a diatom. Here's some kind of filamentous fungal spore. Here's a protozoa, a paramecium, and some bacterial cells, streptococcus. So see, they're labeled as scanning electron micrographs, and the units can be different. So you have to pay attention to the units. Here, really small units, so you probably a good guess that this is some kind of bacteria and not a eukaryotic cell. Okay, so remember we talked about the length of one bacteria was about 1 micron, and that's going to match up about right with that. All right, so let's move on to thinking about classification and identification. Again, a little drier topic, but an important one nonetheless. Let's get some terminology straight. Sometimes we might use all 3 of these terms sort of interchangeably, but if taxonomy really incorporates all 3 of these things. Classification, so putting organisms into a group. Nomenclature, naming them, okay, because we want the name to reflect something about that organism, hopefully, and reflect what other organisms it's related to. And of course, identification, just figuring out which group an organism belongs to. So that should say. Okay, and these are 3 things that is constantly changing as we get more information, as we learn new things about organisms. Organisms are renamed all the time, and they're certainly put into groups all the time, and the scientists don't always agree about what group an organism should fall into. But if we can get that as, I guess as right as possible once a new organism like COVID comes on the scene, well, we knew it was a coronavirus, and that helped us understand, other viruses that novel virus was related to, and so it helped us, understand a little bit more quickly what kind of a disease-causing agent we were dealing with, and what drugs we might have to treat it, or might make it easier to make a vaccine. So it's important to kind of get the taxonomy as close or as good as we can. We talked about Carl Linnaeus last time, and how he's referred to as the father of modern taxonomy. Remember, he did most of his work kind of in the mid-1700s. But he classified organisms based on shared characteristics. That makes sense. That's, he was looking at things that he could see with his naked eye, right? He did some work, and I am not totally clear on things that he figured out, or maybe he just contributed to. But of course, we have, our layers of taxonomy that I wrote in over here that should be familiar to you. Species, genus is species, are smaller ones, and then up from there, family, order, class, phylum, kingdom. And there's actually another layer above this now that we're going to talk to in a minute. How did Linnaeus decide which organisms all belong in the same species? Well, he said groups that interbreed belong in the same species. And that works great for, lions and polar bears, but for bacteria that don't interbreed, that's a bit more of a problem. Okay, now we consider organisms that share about 70% common sequences of DNA to be in the same species. Okay. And with bacteria, we also have, another level here. Strains are basically variations of species where it might just be, some difference in a surface protein, for example. So there's lots of different strains of E. coli, for example. But Linnaeus was really big on naming organisms, and he was first to use binomial nomenclature. That means every organism was assigned 2 names. Okay, and he used Latin. He was Swedish, so I'm glad that he didn't insist that everything was named in Swedish, because that would be kind of confusing for us. So he insisted that every organism have a common name as well as a Latin name. And so with us, we're Homo sapiens, right? The genus is Homo, sapiens is species. Escherichia coli, the genus. We capitalize the genus, that's the way we write it grammatically correct, and we lowercase the species. All right, so get used to writing genus and species, organism names, grammatically correct. If we're going to handwrite them, we underline them, and if you type them, they should be italicized. Now, your book says that you can type them and underline them, and that's still correct. But typically, we're going to italicize them when we type them. Okay, so uppercase, lowercase. Oftentimes you see species in the newspaper or something capitalized, and it shouldn't be. Okay, the genus is typically a noun. Sometimes that's hard to recognize, and most often the species is an adjective. So for example, Escherichia coli, this organism was named for Theodore Escherich. So oftentimes there's a name in there that's sort of made to sound Latin to give credit to whoever discovered it. Coli means of the colon, so that's an adjective. And this is a little bit looser. You don't always see an adjective there, but that's sort of often true. Sometimes I get asked, well, when can I abbreviate and say E. coli? When can I abbreviate the genus? Well, in a research paper, the first time you talk about an organism, you're going to want to write out the genus and species, and then after that, you can always abbreviate the genus. And of course, with this organism, we're most familiar with the abbreviation E. coli. And I just wanted to show this information here because, I got a little bit, okay, ignore this and this that you see here, and that was holdover from something else. Okay, sorry about that little bump there. I wanted to show you, I showed you this in lecture, I didn't post this, but this was from an encyclopedia of bacteria that's over in the library. I don't have a good citation here, but I thought this information was interesting, and I thought it really illustrated why we need to have a good system of classification, because right now it's estimated that there's about 4,000 species of both virus and bacteria, but we estimate that there's a whole lot more species out there that we don't know about yet, that are yet to be discovered. So we need to have a good way to organize all this information that we know about microorganisms. Okay. All right, so Linnaeus, he proposed that there should be all of life should fall into 2 kingdoms, plants or animals. Of course, we've been revising that ever since. And probably one of the more famous revisions of that was the Whittaker revision. When I was in high school in the 80s, this is what we learned: the 5 kingdoms: plants, animals, fungi, Protista, and Monera. Okay, protists were single-celled eukaryotes, and Monera, these were the prokaryotes. This isn't really a term that's used anymore. But this was helpful in some respects. Today, we put even more emphasis on genetic comparisons as basis for classification. I should say classification instead of comparison. And we're looking, the genetic comparisons are going to take into account phylogenetic relationships. And when we talk about phylogeny, that refers to evolutionary relatedness. Okay, we're interested in organisms and their common ancestor in terms of kind of organizing them. Okay, and this study of evolutionary relatedness has actually led to creation of another level above kingdoms, and that's called domains. Okay, this is thanks to some work done by Carl Woese and George Fox. This was fairly recently in the 1970s. They set out to look at genetic relationships between organisms, and they did that by sequencing the ribosomal RNA of many, many, many different types of organisms. Why did they look at ribosomal RNA? Because this is a type of nucleic acid that has a very slow mutation rate. Okay, ribosomal RNA is what makes up ribosomes, and if an organism has a mutation in their ribosomal RNA, then they're probably not going to survive, because ribosomes are where proteins are made, and it's absolutely necessary to the survival of the cell. So a slow mutation rate, and they expected to find that there would be basically 2 very similar groups out of all of life in terms of their ribosomal RNA, that there would be a prokaryotic group and a eukaryotic group. But surprisingly, that's not what they found. They found 3 groups that were very similar to each other. So they decided to call those 3 groups each a domain, and they named them the bacteria, the archaea, and the eukarya. Okay, so obviously these 2, the bacteria and the archaea, are prokaryotes, and the eukarya is the eukaryotes. So here's their tree of life, their phylogenetic tree of life that they published, I think it was in 1990. And I'm surprised this is not in your textbook. But if you take a look at this, okay, you are over here, right? We're part of the Animalia, or animals, and I don't want you to memorize all these names, but in this system, all of these are technically kingdoms, okay, and a lot of them are not recognizable. These are all protozoa. In the eukarya here, the archaea in red, these are going to be even less recognizable, but these are organisms that originally were thought mostly to be extreme organisms, one that survive in high salt or in high temperatures. But we now know that there are archaea basically everywhere around us, just like the bacteria. We even have archaea that is part of our normal microbiome. But we have not found any archaea yet that cause disease. So that's kind of interesting. And here, the bacteria groups, again, these are mostly not going to look familiar. We've got some photosynthetic bacteria. We've got some spiral-shaped bacteria, are gram-positives. These are mostly gram-negatives. We're not really too much worried about these kingdoms. But the really interesting thing that came out of the construction of this tree of life, you would expect the bacteria and the archaea to be more closely related, to have a common ancestor, because they're both the prokaryotes. But look at what we find here. The archaea share a common ancestor with the eukarya. So that was pretty surprising there. You would think that the archaea would be off on this side of the branch, but that's not what we found. Okay, so this is pretty much where we stopped in class today. I'm going to pick up here next time. We started to talk about a few differences between that bacteria domain and the archaea domain. And we'll pick up with that next time in terms of, now we're jumping to chapter 11 here and talking about basically what characteristics we commonly use to classify organisms. So sorry about that goof that I had. Hopefully I'll figure out what went wrong and it won't happen again.